My name is Yanni. I'm an associate professor in the Department of Electrical Engineering of CityU. So today I want to do a brief introduction about my research field. So my goal is to present this so that you can understand my research area. Okay, so I don't plan to go into very deep of these research projects. So the title of my project, my presentation is using deep learning to understand the language of life. So first, let me introduce you what is the language of life. You know the language very well. So for example, a natural language example is English. In this language, we have an alphabet containing 26 characters, and these characters will form words and sentences. So finally, articles. You can make sense out of these English articles. So what is the language of life? So let's use human body as an example. This is a human body, and we are made of lots of cells. Inside every cell, almost every cell, we have almost the same set of DNA sequences or genetic materials. So they are double helix. But if we can stretch this double helix and look at every base in this DNA sequence, that is our language of life. So for example, maybe it looks like this. At the first sight, it looks a little bit of boring because the character or the alphabet is very small. It contains only 4 characters, ACGT. However, the DNA sequence is very, very long. So for example, our human genome has 3 billion positions, roughly. So you can imagine there are lots of combinations or permutations for such a small alphabet. So these DNA sequences, they contain all the commands to tell the cells how to function, how to replicate, etc. So as a result, it determines how we look and whether we have a higher chance to get a disease than others, etc. So it contains the most important information about all the living entities on the planet. So sometimes people call this a blueprint of life. And because we were using computational methods to understand this type of sequences. So this is our language of life. Okay, essentially, they are DNA sequences of all the biological entities on the planet. And not only human, many other animals or even microbes, they all have their genetic materials or DNA sequences. So for example, this is a DNA sequence here of COVID-19, this virus causing COVID-19. So that is SARS-CoV-2. And this was sequenced and assembled by Chinese scientists. And this is the first genome for this virus. And this part shows a timeline comparing 2 epidemics caused by this coronavirus. One is SARS, and the other one is COVID-19. Because of this advancement of sequencing technology, that is how we obtain the language of life. We have now very fast technologies to sequence every base of the DNA sequence. And because of that advancement, you can see nowadays it's very efficient to identify the causal agent behind the COVID-19. Okay, so just several weeks, but back to about 2002, it will take at least half year to know that the causal agent is a coronavirus. So this DNA sequencing technology is closely related to our daily life. Because we have this whole genome, this language of life for this virus, you are able to get your nucleic acid test, because this test actually needs to know the sequence. And because you have the sequence, you can design vaccines, antiviral drugs, and also track the spread of this virus. So for example, you probably know the recent new variant of this virus called the Delta variant. This Delta variant, this genome is actually highly similar to the first reference genome of COVID-19, probably only 15 changes of these nuclear acids. However, its transmissibility is much higher. Okay, so all these analysis applications depend a lot on this type of data. Okay, and now this is really a good time to study genomic data, the language of life, because the sequencing machine, the advancement, has dramatically decreased the cost of sequencing. So this is the cost for the first human genome. And currently we are probably at this point, that is $1,000 per human genome. And the new goal is $100 human genome. So when that happens, this type of a sequencing will become part of your very standard diagnosis test, like when you visit a doctor. So it will have lots of industry and research opportunities. So even with this current cost reduction, it exceeds Moore's law. So that means you cannot just enhance your hardware to analyze the data. You need better algorithms, data structures, computational methods to store, process, analyze, and visualize the data. And of course, all of these data have many different applications in many different fields. Okay, so let me briefly talk about the skills or the tools we use when we analyze our genomic data. So for example, we do need lots of advanced data structures and algorithms, such as how do you store all this data. You actually need very efficient database indexing algorithms to store all this data. And we need graph algorithms. So I talk about the length of the human genome. It's very long, about 3 billion characters. However, today's sequencing machine can only output very short fragment of the human genome, about 250 base pairs. So in that case, every node here is actually a short read output by the sequencing machine. So how do you stitch all these short sequences into the final genome? That needs graph algorithms. And we need some basic statistics and also some learning algorithms such as clustering. So this is a very typical, k-means algorithm, but we also have this is a hierarchical clustering algorithm, a textbook example, but we apply this to the COVID-19 genomes and we can get this type of tree. So this can be used to track the change of this COVID-19 viral genomes. And we also use some learning algorithms, some deep learning algorithms such as GCN and CNN. So I will talk a little more about this part. And we also use lots of sequence analysis algorithms, those decoding algorithms such as the Viterbi algorithm or hidden Markov model. So if you are interested or you are familiar with some of these skills or methods, then you can do some research in this field. So because of the time limit, I will just focus on one type of method we used in a recent research project, that is Graph Convolutional Network, GCN. Okay, so this is an example about our research projects, but in general, we cover different fields. Okay, so let's start from this picture. This is actually an image from electron microscope. So in this image, you can see there are some, green, like with tailed some biological entities, right? And then you also see this golden-ish. Actually, this is a bacteria, and these green entities are viruses, but they are not COVID-19 and those viruses. Instead, these viruses, they infect bacteria rather than human being, okay? So we call them a phage. So this cartoon shows the interactions between the phage and the bacteria. So first, they will bind to this bacteria, just as this image shows, and then they will go into the bacteria and replicate there, and finally, they will kill this bacteria. Okay, so based on this observation, what kind of applications come into your mind? when the biologists observed this, a very useful application today is to use this type of viruses as antibiotics. So you are not probably you have used some antibiotics before. Whenever we have bacterial infections, we need antibiotics. However, today we have more and more bacteria. They develop this resistance to all the antibiotics today. So no drug can kill them. So in this case, this becomes a major threat to human health. One of the top 10 threats listed by WHO. So in this case, if we can use this phage to replace or complement this antibiotic drugs, then this is a very promising application. Okay, so in order to use computational methods to solve, not to solve, but partially address this problem, we have a computational problem. That is, we wanted to identify the relationship between phage and the bacteria. So for example, suppose this one is a new phage, somehow you identified from natural environment, and you want to know whether this phage can kill this bacteria or kill this bacteria. So this is our computational problem. And this problem faces lots of computational challenges. So first, what we know about this type of interactions is very limited. We only know some interactions between a small number of phage and the bacteria. Second, phage is the most abundant and probably the most diverse biological entities on the planet as well. So that means what we know is really, really limited compared to what we don't know. Okay, so how can we identify this type of interactions? So there is an insight, okay, related phages, they intend to infect the same type of host. And then because of that, we want to encode all this information in a graph. So for example, if these two bacteria are related, we end the age. If these two phage are related, we also end the age. And of course, there are lots of specific questions you have to answer. So for example, when I say related, we have to quantify that, right? How do you evaluate the distance between two phage, between two bacteria, etc.? And also, once we have this graph, what do you do? So once we have this graph, we actually use a semi-supervised learning based on GCN to model this interaction inference as a binary classification problem. So in this network, we use GCN to encode all the feature vectors of the phage and the bacteria, and then use that encoded feature vectors as input to a binary classification problem. So basically, my question is, can you tell me how likely these two entities will interact with each other? So this is a very typical binary classification problem. And if you're interested more about this, you can Google our recent publication. So this basically shows the structure of our GCN network, and this is a key insight. Compared to CNN, which you are probably more familiar with, GCN has the advantage of integrating neighboring nodes into this structure. So for example, for the CNN, the structure is the same for all the nodes. However, for GCN, when we conduct convolution, we will only use the local structure, like the neighborhood, either the neighborhood of degree 1, or maybe the neighborhood which are a little farther away. So that is the second layer. So they do have different informations, and the GCN can integrate those topology information and also the node features when we try to understand or predict the link between different nodes. So that's a basic idea about this project. And finally, this is just a summary of my academic experience and also training. So because I got all my training in computer science and engineering, so most of the tools and the methods we use are also from this field. Okay, and after I spent lots of time in mainland China, in US, and finally I joined CityU about 3 years ago. So I think CityU is really a great place for studying for PhD. So I hope you can seriously consider CityU, although I believe you do have many other opportunities. And finally, these are some myths about bioinformatics. So because I focus more on algorithm and tool development, so we actually go very deep in that part rather than just the data processing and calling API. And also, as we focus on more algorithms, we don't use lots of biology. But if you know biology, you will be more sensitive to the research problems. That is always a bonus. Okay, so that's all for this presentation. Thank you very much. And you know how to find me using emails. Okay, so in case you have any questions, feel free to email me.
So hi, Yanni. I saw 2 questions in the Zoom chat.
Okay, so let me see. Now, okay. The first question is, does virus mutation affect the interaction? Yes, it could, because just like how the Delta variant affects its transmissibility, this mutation could also affect the interaction. If you read scientific news, so recently a very big news is a publication of AlphaFold Science and Nature, I forgot which one. So it's about protein structure prediction, right? And sometimes even several bases change can affect the structure of the protein. Similarly, for this virus, if you change some bases, the structure will change. As a result, this virus may not be able to bind to that bacteria. So it will affect the interaction. Yes, a node, there are 2 types of node. One node is a virus, another node is bacteria, but it's not a population. It's one type of virus. So for example, COVID-19 virus, this could be one node, and maybe HBV virus, that is another node. But you can always change your network, depends on your data and the need. So for example, maybe Delta, Alpha, different variants of this COVID-19 virus can be used as different nodes. That's totally possible, yeah.
Okay, thank you very much, Dr. Yanni. And